Hey, good afternoon, everyone, and thank you for your time today. Uh, my name is Mahesh Nair. I'm a faculty member in the Department of Animal Sciences at Colorado State University. And thanks to Drs. Bowker and Kong for inviting me. Um, so this is going to be a slightly different flavor, like Dr. Quach mentioned. You're right, it's not chicken. It's beef. Most of my research work focuses on beef and beef quality. When I mentioned this to Dr. Kwang, he was like, no worries. There are opportunities for learning from other species. So I'm hoping that this talk will spark some ideas and there will be some opportunities for you to learn something new and maybe for me also to pick something new from these sessions. So when you think about meat quality, again, you have a beef carcass there, and there are several things that can affect the quality, right? And one of the things that comes to mind is drip loss. Is that an issue in poultry? Absolutely yes, right? PSC. People have spent their entire career investigating PSC and factors that affect PSC. So that drip loss, that water holding capacity can be a quality issue. Okay, it also affects the profitability, right? When you're trying to sell things, if it holds onto water, it's better. If it's not holding onto water, maybe not. Does color matter if you are making a purchase decision, especially for beef? If it looks like the one that's in the bottom, are you going to pay a premium for that? Probably not. We'll talk about the economic impact of this and the food waste impact of this in the next slide as well. Does tenderness matter? When you are having that product, when you are eating that, you want to have a positive eating experience, especially if you're paying a premium for that product. You absolutely want to have a positive eating experience. If not, they're not going to come back. People are not going to purchase that product again. So one of the quality, I'll discuss two or three of those in this talk, but one of the things I focus specifically on is meat color. Because consumers make purchase decisions with their eyes. When they're walking through the meat aisle in a grocery store and trying to pick up a product, if it looks good, there's a good chance that they will pick it up. More on the other side, if it doesn't look good, they are going to automatically associate that with spoilage. Say, no, I don't want to pick that product up. That's not good. So any discoloration leads to sales loss. So about 20% discoloration, when you get to about some discoloration there, the consumers start to discriminate against that product. And by about 40% discoloration on the surface, consumers are saying, no, we're not going to buy that product. Does that mean it's actually spoiled microbially? Unfortunately, no. Especially in case of beef, the color goes before it's actually microbially spoiled. So that's a big concern. I was part of a study that actually looked at the economic impact and environmental impact of that beef discoloration. So if beef is discolored and it's getting discarded, what's the economic impact and what's the environmental impact? So if you look at the numbers, it's staggering. The average amount of beef discarded due to discoloration is about 194 million kilograms per year. That's about 2.5% of your total beef production. From an economic standpoint, that's about $3.7 billion. Okay? These are animals that are raised typically for beef. It's close to 2 years for their life cycle, right? So these are animals that have been raised for 2 years, that has been processed, put on a retail shelf, and they got discarded because of discoloration. Because it was discolored, they were not picked up, and the stores had to actually throw them out. If you look at the number of animals that's being wasted, that's about 780,000 animals. So that's a huge, huge environmental impact, especially when we are talking about sustainability practices. This becomes a huge factor. Okay? So one of the things that can affect that meat color is the process of muscle to meat conversion. Because muscle is different than meat. And it goes through a early postmortem period, which is short, but it's very intensive changes. Several biochemical changes occurs. So that muscle has to get converted to meat. As you're familiar, one of the things that happens during that time is that glycolysis starts to kick up. The metabolism switches from using oxygen from the aerobic pathway to anaerobic pathways, because there is no more oxygen coming into the muscle cells at that point, right? Because of that, there is ATP that is being depleted, the pH drops, the muscle goes into rigor, and all of that has an impact on meat quality, especially meat color. So there's a lot of biochemical changes going on during that postmortem period. How do we figure what's going on there and what's impacting that meat quality at that point? How is that impacting the energy production and the breakdown of things? So what we know for sure is that what's going on in that early postmortem period has a significant impact on meat quality. It has a significant impact on meat quality. So once we have these quality issues, it's very difficult to revert that. So if we understand what's going on early, then you can build in measures to kind of prevent those from happening. And one of the tools that we have used to understand these is proteome-based tools. So this is very simplistic explanation of how proteomics can be applied. And Dr. Quach gave a detailed explanation of this, so that sets up things nicely for me. So I'm not going to go into too many details. But if you have a protein, you can take kind of two approaches. You can either do a bottom-up approach or a top-down approach. Bottom-up approach is you are basically taking that protein, breaking it down to peptides, and then looking at the peptides, okay, and see if we can build those proteins back up. When you have a top-down approach, you're not breaking down the proteins. You're keeping the proteins intact and ionizing those and looking at that. And this is a very complex workflow of that. But you can use several approaches to this, and you might have come across several of these. It can be a gel-based approach, can be a gel-free approach, can be a shotgun approach, can be a top-down approach. The selection of tool depends on your research question. And this is where it becomes very critical for you to know what's your research question before you venture on a proteomic study. Okay? So I'm going to talk about a couple of studies that have used a non-gel-based approach using peptides. And then I'll also discuss a study that uses purified protein. Okay? That was based on purified protein and specifically looking at what happens to those proteins. So one of the approaches we used for that early postmortem period, like to understand the mechanisms or the protein changes happening during the early postmortem period, was called tandem mass tag labeling. You will see it abbreviated as TMT in the future slides. So again, this is very simplistic explanation of that. Like Dr. Quach might get a little bit squeaky when I butcher this into very simplistic terms. But basically what you're trying to do here is you're trying to run your samples simultaneously. If you run samples coming from different treatment groups simultaneously, it becomes very difficult to identify where did that protein come from. Did it come from treatment 1 or treatment 2? So to avoid that, what we are doing is we are tagging each of those with different colors. Doesn't work simplistically as colors. It could be different isotope labels as well. But then what we do is we run it through the LC-MS program. So front end of liquid chromatography, separate them, and run it through mass spec to both do the quantification and identification. So what you end up with is like a mass spectra like that. You can deconvolute that. You can decompose that. And then from there, you can identify, hey, okay, this amount of protein came from sample 1, whereas this amount of protein came from sample 2. So you can kind of identify where that protein came from. And you can see, hey, which treatments had more of that protein. So it gives an easy comparison across different treatments. Okay? And then you typically do your bioinformatical assays to identify where those samples came from. Okay, so I'm going to use one example from beef, and I'm not going to focus on the details of the results. I'm going to focus on how this method was applied and what are the big learnings we had from those, okay? So strip loins and tenderloins. From a beef quality standpoint, these are two muscles that are very well studied, because strip loins are considered as color stable, whereas tenderloins are considered as color labile, like they discolor pretty fast. So if you put strip loin and tenderloin coming from the same animal on a retail case at the same time, the tenderloins will get brown in about 3 days, whereas the strip loins will stay red for about 7 to 10 days, depending on the sample type. So big difference in their color stability. When you look at tenderness, similar. Like there is some differences in tenderness. Tenderloins are very tender. Strip loins are not something I would consider as a tough meat, but compared to tenderloin, there is some differences in tenderness. So these muscles have been really, really key, like well studied to understand the differences in color stability. So to understand what happens to these muscles during the early postmortem period, what we did was we collected, sorry, I want to explain this. Like if you see PM on the slide, that's tenderloin, psoas major. And if you see LL, that's longissimus lemborum, that's strip loin, okay? So we collected samples from both tenderloin and strip loin at different time points postmortem. So about 45 minutes postmortem, because that's the earliest we could collect after postmortem going through the fabrication process. 12-hour and 36-hour postmortem. And then we did the LCMS analysis to identify which proteins are differentially expressed. We found some protein, like we looked for proteome changes within the same muscle, okay? So between 45 minutes to 12 hours to 36 hours, was there a difference? And then we looked at proteome changes between muscles at each time point. Between strip loin and tenderloin at 45 minutes, was there any difference in proteins, okay? What did we call as difference? When we try to do proteomics experiments, this becomes really critical. What are the standards you're setting? What are you going to call as different? For us, we set the standard as at least 1.5 times difference in expression. For a protein to be considered differential, it has to be at least 1.5 times different, and has to have a p-value of less than 0.05. We also have a standard that we apply internally that it needs to be expressed at least in 2/3 of the proteins. Sorry, 2/3 of the samples that we are examining. So if we are doing 8, it should be at least expressed in 6 of those for them to be included in our results. There are no set standards for this. This is a standard that we apply ourselves to make sure what we are reporting is tight, that what we are reporting is not something that we picked up randomly from the samples, okay? Then we did the bioinformatics assays to look at what are different. Off the gate, we didn't find any differences within the muscle. Okay, between 45 minutes to 12 hours to 36, we didn't find any difference. But there were differences between the muscles at different time points. So this is kind of a summary of that. We found about 71 proteins that are differential between these muscles at different time points. And as you can see, there is a significant overlap between different time points. So I'm going to really run through the next couple of slides because they kind of show the distribution of these proteins. So if you look at the proteins that are different between the muscles at 45-minute postmortem, you have enzymes or proteins related to metabolism, muscle contraction, and fatty acid degradation as the ones that are different. At 12 hours postmortem, there is some changes with mostly the metabolic enzymes taking over. And similarly, at the 36 hours, we will see those are the same category, the metabolic enzymes being the major category. We also did a string analysis just to see how these proteins are interacting with each other. But what I wanted to focus on was the figure that we have generated there. Because this is where we took the data and we examined the data and compared this to the database for about a year, just to identify how those interactions are happening. And that has helped us to generate some big hypotheses on the role of apoptosis, on the differences in that color stability. So when we look at things overall, the anti-apoptotic proteins, they were more expressed in that color-stable muscles. There is a decreased expression of metabolic enzymes and pro-apoptotic enzymes in that striploids as well. So also showed that, hey, we could use this technique to look at the differences in early postmortem period. So I simplified some of the discussions in that paper for the purpose of this presentation. So if you are interested in knowing more details on that, I have provided the reference for that paper. It's published in the Journal of Proteomics, and you can go through that, or maybe we can discuss this offline after the presentation. Okay. Another quality attribute, I'm just trying to make sure I'm on time. So another quality attribute that we discussed, we were interested is in tenderness. Just like color, consumers buy with their eyes, but once they get home and they cook that product, if it's tough, who is getting the blame? Is it because I cooked it wrong, or is it the company that sold me the tough meat? It's always one person that gets the blame. Other than mom, it's always the fault of the person who sold that product, right? So if it's tough, we have a problem. The repurchase decisions are based on that eating experience. So if you are going to buy that product again, if you're going to go pick up that product again, is going to be depending on that tenderness. And tenderness is determined by the proteolysis, breakdown of proteins during that postmortem period. So proteins that are intact gets broken down during that postmortem period. And one of the enzymes that plays a significant role in the postmortem proteolysis is called calpain-1. So these are calcium-activated proteolytic enzymes, and those are largely responsible for postmortem proteolysis. And they sometimes stay active up to 6 days. Most of the improvement in tenderness happens during the first 12 to 24 hours, but they can remain active for slightly longer time. The challenge is that muscle tissue contains mono and polyunsaturated fatty acids. Okay, and unsaturated fatty acids are susceptible to oxidation. When we have oxidation, what do they generate? They generate aldehydes or ketones. And these aldehydes and ketones, they can form covalent bond with proteins that are present. Okay? So oxidation happens. Oxidation has a byproduct. That byproduct can affect other proteins that is in the system. Okay? So the reactivity of these aldehydes depends on the number of carbon atoms present. So these are some of the common aldehydes that we use in vitro systems to look at the impact of these aldehydes on proteins. Okay? So if you see malone dialdehyde, abbreviated as MDA, that is a 3-carbon aldehyde. Hexenal is a 6-carbon compound, and 4-hydroxynonenal is abbreviated as HNE, is a 9-carbon compound. Typically, I use the word typically in quote, typically their reactivity is depending on the number of carbon atoms. So from low number of carbon atoms to when it goes to high number of carbon atoms, their reactivity increases. With me so far? So we have done studies that have looked at the impact of these products on different enzymes. Okay? So we looked at the impact of these hexenal malone dialdehyde and HNE on lactate dehydrogenase and NADH-dependent reductase systems. You can see that, hey, compared to control, there is difference in reactivity. So we wanted to see their interaction with calpains and how can they affect their stability. Because calpains, they are susceptible to oxidation. Studies have shown that if you expose them to reactive oxygen species, they can inhibit calpain function. So if there is oxidation in the system, that could affect calpain function. Also, if you have high oxygen in the packaging, like high oxygen modified atmosphere packaging, the meat tends to be tougher in those packaging compared to aerobic traditional packaging. So that presence of oxygen, which initiates that oxidation, has an impact on calpain and that tenderness. So we wanted to see how these products, that MDA, which is 3-carbon, hexenal, and HNE, which is 9-carbon, affected calpain activity, and what happens to the calpain structure. So we designed a simple experiment. This was done in collaboration with Doctors Elizabeth and Stephen Lonergan from Iowa State. They have a system where they isolate calpains and purify calpains from pork muscle tissue. So again, it's a different species, but once you isolate to protein level, they have same sequence, same structure, so we can use that across the species. So what we did was, we incubated these calpains with these 3 different oxidants, so MDA, hexenal, or HNE, at 3 different concentrations. Gave them about 24 hours at lower temperature to react, and then looked at their activity. What happened to the calpain activity when we exposed these oxidants? What we also did was, we also looked at the changes in calpain with the exposure to these products. So what happened to calpain structure? Did these compounds attach to calpain, and looked at that? Okay. So when we look at the activity, to our surprise, when we had MDA, there was not a significant change. Like there was a slight increase in MDA activity, in calpain activity with 100 micromolars of MDA. But there was no difference in other concentrations. That was a surprise for us. Might be explained by some of the things that we saw in the adduction sites, which I'll get to in the next couple of slides. But when we looked at hexenal, yeah, we saw a concentration-dependent effect. As the concentration increased, the activity decreased. The calpain activity decreased. When we looked at HNE, same. Like as the concentration of the oxidant increased, the activity decreased. Okay? So what we wanted to do was to look at the structure and see where these proteins are getting attached. Okay? So we looked at the structure of calpain. Calpain has a catalytic unit, which is represented in that greenish teal color, and has a regulatory unit that's represented in that pink color. Its active site has histidine, asparagine, and cysteine in there, and that's actually represented there. So we wanted to look at, okay, when we expose to these oxidants, what is happening to these products? This is where we got a little bit of a surprise, because we had controls and samples that were exposed to oxidants. But when we are looking for adducts, we found adducts in the controls itself. So we found MDA attaching to different sites on calpain controls. Remember, these were not exposed to any oxidants. So what this shows is that there was oxidation happening in the system already, and these are getting already bound to these oxidants are getting bound to the proteins automatically, even without us exposing to these oxidants. Most of those in MDA, we found it in the catalytic subunit. We think that might be the reason why there was not a change, or there was a slight uptick in activity with the MDA exposure. When we look at the HNE adduction, similarly in controls, we found some adductions in HNE as well. Okay? When we looked at the MDA adduction sites on the samples that were exposed, you can see that there was adduction actually on the active site. So MDA bound to the histidine that's present in the active site. When we look at the HNE adduction, again, you can see that there is adduction happening in asparagine, glutamine, and histidine. So this is the first study actually showed that, hey, lipid oxidation products can have a differential impact on calpain activity in a concentration-dependent manner. And when we incubated with hexanal and H&E, there was a decrease in activity. And we found adducts, both MDA and H&E adducts in the system. The bigger question that we came out from that project was the adducts that were present in the control. So we're going back to the drawing board and say, okay, where is this happening? Because when we isolated calpains, these were isolated from muscle tissues that were going through a slightly postmortem process. So they were already happening in the system, and we need to identify when and how they are happening. Okay. Again, that study is published in Journal of Ag Food Chem, and we did a follow-up study on those using calpains too as well. So both of those you can look up and find more details in there. I'm going to really quickly talk about a slightly different approach, and this is not a proteomics approach. So this is a slightly different tool that we are using to predict tenderness, juiciness, and flavor of beef muscles with different aging times. This tool is referred to as rapid evaporative ionization mass spectrometry. It's a mouthful, so we have abbreviated that as REAMS, and that's how it will be referred in the future slides. So what does REAMS mean? This is a tool that we can use for in vivo, in situ, real-time identification of biological samples. Okay? So this was developed for actually using in human surgeries to distinguish between cancerous and non-cancerous tissues. And this big difference here is that front end, like the back end of that is a mass spectrometer. The sampling device is where the difference is. It's called as an eye knife, which basically cauterizes the sample and generates a fume that goes into the mass spec. Okay? It generates a fume that goes into the mass spec that gets cleaned up, and then it gets ionized and gets fed into the mass spectrometer. Okay? When you look at the samples, this is how that sampling device looks like. You're basically burning the samples to generate a fume, and you are analyzing the fume that's being generated by that. Okay? The best example or the best analog I can come up with is it's like soldering iron. So it's basically burning through that meat product, generating a fume that is analyzed there. So it's a relatively new platform that can capture molecular fingerprints. Okay? The good thing is that it provides a real-time mass spectra. There is no sample prep. You don't need to prepare the samples. There is no extraction, like Dr. Bowker was asking earlier. That can be a challenge, what kind of extraction we are using. So there is no sample prep. This can be real-time. The acquired mass spectra can be then used to develop statistical models. And this is where it gets a little bit tricky. So I'll get into that in a minute. But how does that data look like? So this is how a simple way to look at the data. So this is basically the chromatogram coming from there. So each of those peaks is when you are burning that sample. Okay? So typically what we do is we burn the sample about 5 times so that it's a little bit more representative rather than a single burn. So each of those peaks represents a burn. And when you deconvolute that further, you can see the mass spectra. The challenge is that this spectra is not annotated. So there is no annotation on the spectra. So you cannot identify what is what. Instead, it generates a molecular fingerprint. So if I'm taking samples from tenderloin, this is how the spectra will look like. If I take samples from a strip loin or from another muscle type, this is how this spectra will look like. Okay, so it creates molecular fingerprints. You can generally say, hey, this spectral region belongs to fatty acids, this spectral region belongs to, sorry, I'm trying to keep track of time, phospholipids, okay? So what we wanted to do was to look at the ability of this machine to predict eating quality after different aging periods. So 3, 14, or 28 days aging. We did the shear force assay, so both sliced shear force and Warner-Brasler shear force, which are common methods used in beef, and then did a sensory panel to identify the flavor. When we collected the samples, we also collected samples at 36 hours postmortem. So before any aging, we collected samples and subjected those to reams. The idea was that, can we use this 36-hour sample to predict the quality after 3, 14, and 28 days of aging? Okay, if we can do that, before we send the product out, we know the quality. Okay, so that was the premise of that. We did the sensory analysis and did a principal component analysis and separated the flavor attributes. Okay, so most of the flavor attributes you associate with beef, like fat, like buttery, umami, they all got separated from the typically negative flavor attributes, like liver, sour, and bitter. These are considered negative flavor attributes. They got separated. We also further separated them using a hierarchical clustering into acceptable and unacceptable. So if it's in orange color, it's an acceptable sample. If it's a teal, it's unacceptable samples. Okay? Then we used machine learning algorithms to see if we can predict the quality based on the reams data we collected. And there's a lot going on this, so I'm going to take a minute to orient you to this graph. So on the x-axis, you have that percentage accuracy. Okay? How accurate is the reams prediction? And on the y-axis, you have different quality attributes. So SSF is slice shear force, Warner-Brasser shear force, sensory tenderness, juiciness, and flavor. Okay? D3 in orange, D14 in teal, and D28 in green. Okay, I'm going to pick one of these. So when you look at slice shear force, all of the models they had at least 80% accuracy. So we were able to predict it with at least 80% accuracy that, hey, this sample will be tender, this sample will be acceptable. Some of them even went up to 90 in different attributes. Okay? I presented the very positive, very brief part of this study. We ran into some limitations and challenges on this study, especially when the samples were overlapping. When they had slight overlap in categories, it became very difficult to separate. So you can, this study is published. You can look this up online, and that goes into a lot more details regarding how we did the machine learning algorithms and developed the models. Because that was the most critical part of this study, was how we developed the machine learning algorithms to analyze the data. Generating data is one component. How you analyze the data and how you interpret the data is, for me, the most important component of any of these studies. So just some concluding thoughts. Oxidation happens in a system. Like we found adducts with calpains when we are not looking for them. We were not expecting them to be there, but there was oxidation happening in that system. And what we have found is all of these quality attributes are interrelated. So when you talk about color and oxidation, that oxidation can have an impact on color. That oxidation can have an impact on flavor as well. So we need to take a step back when we are thinking about this and take a little bit more of a systems approach to do this. And make sure you're using the right tools based on the research question you have. So that selection of tools, which kind of approach I'm going to use? I'm going to use a peptide-based approach or a protein-based approach or a totally different approach is going to be driven by your research question. So I think I kept that within the time limit, and thank you for your time. Appreciate that.